Hi everyone, today we'll be going through GCSE Edexcel Biology Predicted Paper 2 for 2024. Please still revise every topic since no one will know what comes up on the day. Also subscribe and like and turn on your notification bell because we will be doing more predicted paper for Chem and Maths. I also have linked timestamps in the description for more specific topics as well as access to the PDF copy, and also linked a couple more videos that I think will be useful for tomorrow's exam. Let's get started. Question 1. Read the passage below and use the information in the passage and your own knowledge to answer the question that follows. Snail farming. Usually in an exam setting, I would recommend that you do this question at the end so you really do have time to read all the passage correctly. However, for the sake of the video, we'll be doing this question first. And we'll be skipping to the questions directly, though I recommend that you read the whole passage before you go into the question so you get an idea on what they're going to be asking about. So A, explain why snail meat is healthy to eat, and they are telling us to look at line 4. So if we go to line 4, we can see that it says, the meat itself is in high protein. It's also low in fat, so snail meat is healthy to eat. So they already told us that it is low in fat. This is really good because that means there's less risk for heart disease or blocks blood and vessels. Also, they did say it was high in proteins and amino acids. This is also very good because it's very good for your bodies, because for growth, repair, for our muscles. B, suggest why conservationists are pleased with that snail farming exists. Now, if you see in the next line, they say that also snail farming helps to conserve natural snail population, which is at risk due to collected in large numbers in many places. This means that they are happy because it conserves snails, and because of that natural population, thus is collected so that not many, so that we are able to conserve them. C, name organisms used in biological control in snail farms. If you see over here, it says natural predators of the insect pest are spiders, which are encouraged as farm. So these are spiders. D, the shell of a snail is called an exoskeleton. Use this information to suggest one mineral ion on the diet of a farm snail that would help them make this shell. They say it is a skeleton, so it's just anything that will help bones grow, so we know calcium, or also it could be phosphate. E, I suggest why many organisms can assimilate green vegetables very well. Lines 14 and 15. So 14 and 15, green vegetables are not assimilated very well by many organisms. The assimilation energy AE organism of percentage total food eaten is absorbed by the blood after digestion and not lost in feces. Carnivores tend to have AE of 80%, and most leaf-eating herbivores have around 50%. However, snails have a symbiotic microorganism in their gut that releases the enzyme cellulase to digest cellulose in glucose. This boosts their AE to at least 75%. So because why many organisms is not, they kind of gave us a clue that it's something to do with cellulose. So most of them cannot digest cellulose, they cannot break it down, and this is because due to the lack of enzymes, they do not have that cellulose that what a snail does have, so there is no absorption of that cellulose and fiber. 2I, calculate the assimilation efficiency AE of a snail that ate 4g of food and 1.2 of feces. So if we saw on our paragraph, we can see over here that they say that the assimilation efficiency are organism in a percentage of the total food eaten, not as loss as feces. First thing we are going to do, we're going to minus them from each other. We do 4 minus 1.2, and this equals 2.8. Now we want to see the efficiency, so that means they want the percentage. So overall they ate food, so 4 would be in the bottom. However, only 2.8 of it is actually given because this is not lost in feces, so 2.8. So we divide them together and we times it by 100, and this means our answer will be 70%. F, suggest why production efficiency PE of birds and mammals is much less than PE of snails. The 23 and 24. So over here, the production efficiency of an animal in a proportion of assimilated food is turned into new biomass rather than being metabolized. Birds and mammals have a very low, but snails have much higher, and this is because metabolic use of assimilated food, particularly in glucose, is much reduced in snail. When they talk about glucose, we are talking about metabolism and respiration. So our answer is that there is more glucose metabolism and respiration, and also this means that we have less heat loss is going to be needed because needed to maintain that body temperature and keep warm. That means they are more active, so snails, they're able to conserve that and use less energy. G. What is meant by the line term selective breeding? Line 26. Line 26. Selective breeding is used on a snail farm to produce fast-growing snails that have thin shells and lay a lot of eggs. Selective breeding is basically when humans actually select their desired characteristics for breeding and they breed those two together so that they can get whatever they want. So in here, they wanted fast-growing snails that have thin shell and lots of egg. So as humans, we will choose those snails that have those specific features and we breed them together so that their offsprings will have these features that we want and all the population in turn will have that characteristic. Suggest why snail eggs are put on soil that has been sterilized. So line 27, this is the eggs are put on damp, sterilized soil, incubated. The young snails are put in hatch in a plastic tray as they're easier to clean. We just don't want any bacteria, microorganism, fungi, pathogen, infection, or disease to affect them. 2. The passage describes how a virus can affect humans. Complete the passage by writing a suitable word or words in each of the spaces. Humans are affected by many viruses. Viruses only contain one sort of, this is nucleic acid, either DNA or RNA. One virus which causes the disease AIDS is a HIV virus. And people with AIDS are vulnerable to infection because their immune system does not work so effectively. This means that they are unable to produce white blood cells that normally fight off infections by producing specific proteins which are called antibodies that help destroy pathogens. Diseases caused by viruses can be prevented by injecting the body with an inactive form of virus. This method of injection is known as a vaccination and stimulates the body's defense system to produce memory cells. 3. The diagram shows 3 duckweed plants in a beaker of distilled water. Explain how this water moves into these plants. So we have these water over here and here are our plants. So water moves in by these roots over here. They move in by the root hair cells and this is through a partially permeable membrane. We're talking about water. So how does water move? They move by osmosis and this is down a concentration gradient. When you're always talking about a substance being transported, make sure you always say from what type. This is from a partially permeable membrane. What is the surface they're moving from? How exactly? Osmosis, diffusion, active transport. And always put down a concentration gradient, or for active transport, it will be against a concentration gradient. This alone will give you 3 marks. Salt was added to the water in a beaker to form a salt solution. Explain the salt solution would affect the movement of water into and out of the plant. Because it is put into the beaker to form, this means we're going to have less movement of water into a plant, and this is because we are going to have more water moving outside. It will be moving out of the plant because there's now a salt solution, so they want to make sure that the concentration of water is the same in both sides. So that means because less water outside the plant than inside, so it will go into it so that it can dilute it because it moves down a concentration gradient. Complete the sentence by putting a cross X in the box next to your answer. When concentration of mineral ions in the soil is greater than the root hair cell, mineral ions are transported by the root hair, and this is by. So the concentration in the soil is greater, so it's going to move in, and this is down the concentration from a high to a low, so this is diffusion. B. Explain how water, glucose, and mineral salts are transported through a plant. We are going to split this up for each one. So it is 6 marks, so try at least to put 2 points for each type. So with water, it is transported through the xylem, and this means that osmosis into the cell of the leaves, and also there is evaporation from the leaf, and that's what causes the transpiration stream, and diffusion into the atmosphere, and this is through stomata. Now we have our glucose. This is converted into sucrose in a plant, and it's dissolved in the water. Then it's transported through a phloem, and this is bidirectional, so it moves both up and down. Now we have mineral salts. This is dissolved in water and then it moves through the xylem, and this goes from the root to the tip. Like how we said before, we talk about transport. We always say from where to where does it go, or how exactly is it transported. Here we say xylem, phloem, and xylem. We say how it's moved. In this case, we say if it's dissolved in water, or we could even say if it was diffusion, osmosis, or active transport. 4. The diagram shows the human circulatory system. This is the blood vessels in organ X, the blood vessels W, and blood vessels Y. Name the organ X. So if you follow it, we know X is over here. So where did it come from? It came from this dark over here, so it came from this side over here. We know that this is basically what the body, so it goes back, and what it does is that we want to go ahead and oxygenate all this deoxygenated blood that we just came back from the body. So this blood vessel here, see, it goes back to the heart, it doesn't go anywhere else. We can see that this is the lung. Explain how the heart causes the blood to move to organ X. So what does, we can see that the blood first moves to the right atrium, and then it goes to the right ventricle. And then how it goes up is that the heart contracts, so that means the blood pressure increased by the heart, so it pushes the blood so that it can move into the pulmonary artery. Which rows show the name of the blood vessels W and Y? So we have W over here and Y over here. This one's going and this one's going out of. So this has oxygenated blood, this has deoxygenated blood. So it is D, we have vena cava and we have aorta. Describe how blood vessels in W is different from blood vessels in Y. So we know that blood vessel W is the vena cava. So this means there is a much lower pressure. This is because there is a lower concentration of oxygen, meaning a greater concentration of CO2. Also, it is also darker. It's dark red. B. The diagram shows the chambers of heart. Calculate how many times thicker the wall of chamber B is compared to the wall of chamber A. So what we would do is we would measure this over here from where they told us and this over here. And then you would divide our width of ventricle wall B, and then we divide that by the measure of the width of ventricle wall. And you would see that it is going to be 3 times bigger, any number between 2.5 and 3. It's going to be different from when I do it on here because I'm doing this electronically, so the sizes won't be the same as if you're doing it in paper. Explain why the wall chamber B is a thicker wall than chamber A. This is because the wall is much stronger, it has more force, more powerful, and this is because the blood is from the left ventricle, so it's under a much higher pressure than it is from the right ventricle because the blood needs to be pumped all the way through the body, not just to the lungs. Question 5. The diagram shows one of the stages of the nitrogen cycle. Complete the sentence by putting a cross X in the box next to your answer. This stage is, so it goes from ammonia in soil to nitrates in soil. This is nitrification. Explain how plants use nitrates from the soil. What they do is nitrates are used to make proteins because there is nitrogen in those proteins, in these amino acids, and we use proteins for growth. So this is a 2 marker, so we need to put 2 points. Describe how an overuse of nitrate fertilizers can cause eutrophication. So this is the same thing over and over again. It's the same structure. The first thing that we say is that nitrates leach into the water. They go into the water. And this means, let's say if we draw it out here so that we can make it more. First of all, we have our nitrates that go into the water. And because our nitrates go into the water, we are going to start see this algae start growing rapidly. And this tend to grow in the top of the water right on the surface. There is plants that are down in the waterbed. These now aren't able to get the sun that comes from it. There is no sun that can go because this algae now that just grow rapidly is covering the sunlight from being able to go in. So these underwater plants can't photosynthesize anymore. They can't make their energy and their food because sunlight isn't getting to them. So this means they are going to die. And once they do die, we have these decomposers and bacteria that come on and they feed and they break down this dead end plants. And these bacteria start using up these oxygen that is already in the sea. So because they're starting to use this oxygen, any fishes that are in the sea are going to be dead because there is a lack of oxygen now within this water. Leguminous plants such as beans and peas have bacteria growing inside the nodules of their roots. And the diagram shows the nodules on this root. Explain the relationship between this bean plant and the bacteria growing in root nodules. We have our root, and these little small things are our bacteria. So what it is that bacteria uses these nitrogen, and it's nitrogen fixing bacteria, and they make ammonia and nitrogen compounds, and these are going to be used by the plant. And this is so that we can use proteins and make proteins for growth. And then the bacteria protected is within the root. And then bacteria instead is going to obtain glucose and sugar from the plant, and this together is called a mutualism relationship because they all get something from each other. They basically take because they all take something from each other. 6. The diagram shows a piece of apparatus called a calorimeter. The calorimeter is used to investigate the energy content of 1 gram of glucose, and the glucose burns in a calorimeter and energy is released from the heat of this water. Suggest why oxygen is passed in the calorimeter. This is because it needed for combustion and burning. Oxygen has to be there so it can continue the burning going on. Suggest why the waste pipe is coiled. So we can see over here it is coiled. This is because it's going to increase its surface area, making it more distance and more time. So there's going to be a better heat transfer to the water, so it's heating the water more and at a much higher temperature. Suggest why stirring the water will improve the data collected. This is because it distributes the heat, spreading it out, so it makes all the water the same temperature, so that is controlled. The calorimeter contains 200 grams of water, and the graph shows the rise in temperature of water in the calorimeter during this investigation. Use the graph to find out the increase in temperature of the water during this investigation, how much it increased by. So we take in the first and the bottom. So over here we can see it goes up by 1, so this is 16. And over here this is 23, so over here will be 23.5. We do 23.5 minus 16, and that's going to be 7.5. Use the formula to calculate the energy content of 1 gram of glucose, where its energy content in G is mass of water in grams, temperature increase, times 4.2, and mass of food. So we have the mass of water, this is 200, times by the energy increase, that's times 7.5. We times this by 4.2, and this is mass in food, where they say over here it is just 1 gram. So if we do this, we will get 6,300. Question 7. The diagram shows a section of an eye with iris parts A, B, and C labeled. Name parts A, B, and C. So A over here, this is our lens. And then we have B over here, this is our cornea. And we have C, which is on the back, and this area is called the retina. B. When you move from a bright room to a dark room, you can't see very well for a while. After a brief time, a change in your iris helps you see more clearly. The iris contains a muscle tissue. What is meant by the term tissue? It is similar cells that are all working together to perform the same function. Describe the changes that take place in the iris when moving into a dark room and explain how they help you see more clearly. What happens is that radical muscles contract and the circular muscles are going to relax. So this means the pupil dilates, which means that the iris gets smaller. And this just means that more light can enter, therefore we can see more clearly. 8. The diagram shows a structure of a female reproductive organs. Name structures labeled in the diagram. First we start with P. This is the oviduct or the fallopian tube. And then we have Q over here. This is the ovary. R is the uterus, and S is the vagina. Describe the changes that take place in structure R during the menstrual. Structure R is the uterus. So first of all, it grows and it thickens, and then after it remains the same size for a while, but then it breaks down in shedding, and this is during a menstrual cycle. However, it is not broken down if the pregnant or if the egg is fertilized. C. Some women only have sexual intercourse at certain times in their menstrual cycle to avoid pregnancy. Explain why this is not a reliable method for birth control. This is because ovulation may vary between one woman to another woman. This means that it can ovulate on different days in each month, so the cycles can be irregular. This means that the sperm can survive during that time for a longer. Describe the role of estrogen at puberty. This is responsible for the secondary sexual characteristics. So it starts at menstruation, and there's lots of different characteristics such as hips widen or the growth of body hair or the change in distribution of fat. The diagram shows an animal cell called a neuron. Name 3 structures found in a plant cell that are not found in a neuron. These are all the same because this is an animal cell, so we can just take it so that it's a cell wall, chloroplast, and a vacuole. B. The neurons are involved in a reflex arc that helps humans respond to a stimulus. The diagram shows a reflex arc labeled A, B, C, D, and E. The arrow shows the direction of the nerve impulse. Complete the table by naming each part of the reflex arc, and one has been done for you. So A is over here. This is the receptor, what detects the change in the environment. Then we have C. C is over here. We can see that this is in the spinal cord. So this over here is our relay neuron. Then we have D. This is carrying out the effect, and it's going to this over here, which we can see is a muscle. So that is the motor. And that muscle we just saw is going to carry out the act. So that is the effector, or you could just say muscle. There is a small gap in between neuron B and C. What is the name of the small gap? This is a synapse. Nerve impulses can travel along a neuron at 120 meters per second. The distance between a spinal cord and a foot of a human is 90 centimeters. Calculate in time seconds what it would take for the nerve impulse to travel from the spinal cord to the foot of this human and show your working out. First, we know it's distance that is over speed and time. So we are trying to get time. So what we do is we do time, and this is over distance over speed, like in how our triangle shows. Because our speed is in meters, we're going to turn this 90 centimeters into meters, so it would just be 0.9 because it's divided by 10. So we do 0.9 and we divide that by 120. Our answer will be 0.0075 seconds. 10. When bacteria divide, they replicate their genome and synthesize their cell wall. Figure 12 outlined the stage of bacterial replication. So we have our cell wall, plasma membrane, a double-stranded loop of DNA. They replicate, then they divide into two. Penicillin inhibits the synthesis of cell wall in bacteria. Explain the effect of penicillin in bacterial and human cells. So over here, they gave us a very important information that it affects cell wall in bacteria. So first, we can say that penicillin prevents bacteria from dividing because they do not allow them to make a new wall. So without having that new wall, they can't make that new bacteria. However, humans are going to be completely unaffected by it. This is because human cells, none of the cells in the human body has a cell wall. B. Penicillin, isolated from a fungus, was the first antibiotic used to treat bacterial infection, and it's still widely used today. Scientists have genetically engineered bacteria to provide a large amount of penicillin. Describe how scientists could produce a genetically modified bacteria that produces this penicillin. What they do is they first use a restriction enzyme. This is to remove the gene and cut the plasmids. And then they use ligase, and this is joining DNA and molecule together, kind of like glue. Then you cut the gene from the genome of the fungus and extract the plasmid from our bacteria. Then you insert our recombinant plasmid into the bacteria. C. MRSA is a bacterium that has evolved to become resistant to antibiotics. With reference to Darwin's theory of evolution by natural selection, explain how MRSA bacteria has evolved to be resistant to antibiotics. With natural selection, it's always the same structure. We just need to adapt it to our stimulus that they give us and apply it. So first thing we can say is bacteria reproduce rapidly, generating a very large population. So there was a variation among a bacteria population, and some bacteria has developed this resistance to antibiotic, and this is always through mutation. Then antibiotic treatment is going to exert a selection pressure, and because of this, bacteria resistant to antibiotics are going to survive, and these antibiotics will be passed on because they will be able to reproduce, and therefore it is inherited and the allele has been passed on to the further generations. However, the non-resistant bacteria do not survive, so they won't be able to pass on their genes. This means that level of antibiotic resistance in a population of bacteria is overall going to increase. 11a, the diagram shows one strand of DNA of a section of a gene. What could be the complementary sequence of bases on the messenger RNA produced from this strand of DNA? Because it's RNA, remember all the T is going to turn into U. So we're just going to put our opposite. Here it would be T, so instead we're going to make it U. The opposite of T is A, the opposite of C is G, then we have C, then we have U, C, A. Now we got to just cross-reference, and it is C. This diagram shows a stage during protein synthesis. Which rows give the correct names for the stage of protein synthesis and show the sequence bases labeled X? X over here is our anticodon, so we know it's not B and we know it's not D. This shows translation because transcription is the first thing that we just did over here, which is copying DNA to RNA. C. Explain how a mutation in a gene can affect the phenotype of an organism. So mutation is rare and random in a change in the DNA. So what it first does, it changes in the nucleotide. So this means it changes bases and triplets either by causing a frame shift or by substitution. So this means that there's going to be a change in the sequence of amino acid. This means it changes the secondary structure as well as the tertiary structure. So there's going to be a change in the enzyme and protein, therefore they would change in the function, therefore changing in our phenotype of the organism. D. In an accident at a nuclear power station, radioactive material was released to the surrounding areas of land, and scientists investigated this impact on a species of butterfly. Samples of adult butterflies were collected from an area near the power station 2 months after the release of the radioactive materials. The percentage of butterfly with abnormalities were calculated, and the butterflies then were mated in the laboratory, and the percentage of offsprings with abnormality was calculated. The experiment was repeated with butterflies and collected from the same area 10 months after the release of radioactive material. I. Describe how the scientists could have sampled the populations of butterflies in this area. First thing they do is you are going to use a quadrant, and then they grid the area so that it kind of looks something like this. And then what they do is that they randomly select different areas. So let's say they'll say A and B and C and A. And then you randomly do this either by a number generator or picking it out of a hat. And then you use your quadrants and you put it into that section. You then count the butterflies, how many butterflies are in that area. And then you repeat it multiple times and you calculate a mean. 2I. Results of the investigation are shown in the table. The scientists concluded that the increased level of radioactivity has led an increased rate of mutation of DNA. Discuss the scientists' conclusion and referring to the data from the table to support your answer. So if you look at the table, we can see that 10 months after the accident, there is much more in both ways. However, there is more in our offsprings than there is in the adult. And when they say discuss, they want both the positive and negative. So we want to say why we support that conclusion and why we also disagree. So what we can say that increase in abnormality butterflies, there was more abnormality butterflies 10 months after it. And this is probably due to a long exposure of radioactivity. Also, there is a much larger increase in number of abnormal mutated offsprings compared with the adults after 10 months. We can also give a reason on why we think this is. This is because mutations and it's passed on to the offsprings. So recessive mutations may have been carried by these adults. And also other factors can cause these abnormalities. So it might not always be due to genes and DNA. It could be due to that radioactive. So there is no control experiment, however. So this is our against. There was no control experiment. So we do not actually know how much of this is actually due to radioactivity. Statistical analysis will be needed. And also our radioactivity was not measured, so it may have changed, there might have been decay, and we don't also know the number of butterflies that are being sampled. That is the end of the predicted paper. If you have any questions based on this predicted paper or content in general, just put it down in the comments. I'll be more than happy to help. Good luck on your exam tomorrow. I hope it goes well.